Good. Hello, everyone. It's a pleasure and an honor to be here presenting my work. And in the interest of time, I will just focus on the main ideas and gloss over some of the technicalities. But if anyone is interested, I'm always happy to discuss later. So this talk is about macroproteins. And more in particular, what I refer by macroproteins are single multi-domain massive proteins in terms of length. You have from few thousands of amino acids to several thousands of amino acids. They play a lot of different roles, and most of them are located on muscle cells. Here you have a schematic representation of the sarcomere, which is the functional unit of the muscle. And here you have highlighted one protein in red, titin, which is a protein that our experimental collaborators and our group is particularly interested in, which is the biggest protein in human, made of 35,000 amino acids. And to give a feeling of how big really titin is, here is a schematic representation of all the domains that titin is made of. So each of these orange lines represent an immunoglobulin 1 set domain, and each of these green lines represent a fibronectin type 3 domain. So it's a really massive protein. And as you can imagine, when you try to deal with proteins as big as this one, you start to run into issues with the conventional methods that you could apply to other proteins, both computationally, but also experimentally. And one of the common solutions, or one of the few solutions, would be to chop it up by domains and try to study that way, and then try to do some inference of their properties. So for example, if you would like to model this with AlphaFold, you will need to do it by parts and then try to find how to stitch it together. If you want to do molecular dynamics or other physics-based methods, you would also need to break it into parts and then try to find how to combine this data. And experimentally, if you want to do biophysical characterization, for example, thermal stability or things like that, you will also need to express it by domains because the protein itself would be too big. So in the field of variant prediction, we are interested in using protein large language models, and have already heard in a lot of talks. They have been showing an unprecedented performance in a lot of fields. Also in the case of variant prediction, either from a zero-shot kind of usage by just looking at the probabilities, or as part of a more complex pipeline by either using it as part of a pipeline or using the embeddings as features for other models. However, we were worried that maybe these models could underperform on domains because they were trained on mostly full-length proteins. So more in particular, the data set that it was trained on, in the case of ESM, contains 90.3% roughly of full-length proteins. And this was motivated by a very known behavior of these PLLMs, that is that the first position is almost always predicted to be a methionine. And the reason why is because all full-length proteins start with a methionine, because that's the amino acid that is encoded on the starting codon, but not due to a biophysical property that we would be interested in. So when we are showing domain sequences to a large language model like this one, we don't want the first amino acid to always be predicted as a methionine. And of course, you could add a methionine at the beginning and then just treat your first amino acid as the second one, but we were worried that maybe this bias that is present on the first position could also be applied on other positions due to how these transformers encode positional information. So what we did is for the domains of titin, so fibronectin and IGs, we went to Interprot, we downloaded sequences with annotated that had annotations of these domains, then we chopped out these domains and we showed to the model either the domain alone or the domain with the full length context. And here we are trying to see discrepancies, so how different are the predictions in terms of probability at those positions. And here you can see it in the plot. So the plot is cut because the first position goes really high up. But in general, on all the positions of the domain, there was a consistent under prediction. So this is the ratio of the prediction using the whole context or only the domain. And the line is over 1, indicating that in this case, all the positions were predicted with less confidence for the case of the domains. And up to some degree, this is expected because you're removing context. So you would expect the model to be less confident. But if you think of domains, they are defined like a functional unit, you would expect a lot of the evolutionary pressure that these models can pick up to still be present. So you would have expected the average difference between the predictions to be very close to 1. And this is not the case. And here on the right, you can also see a distribution of these discrepancies, showing also a skewness to the right indicating that domain are under-predicted. So we wanted to see if we could correct this under-prediction or bias for the domains. So we did this kind of pipeline. So you start with your target protein, in this case, titin, which is your protein of interest. You scan for the domains. You find the different domains. Then you go to Interpro. You download sequences with those annotations. You chop them up. You do dataset curation. And then you can fine-tune a model based only on these domains. And we fine-tune for very short, only one epoch, to avoid a kind of overfitting known as catastrophic forgetting. So here are the results after this fine-tuning, which is the green line. This is the same experiment of showing the whole context or only the domain. And what you can see on this green line is that now the predictions are equally confident when you have domains or whole-length proteins, and also the distribution became narrower and more centered at 0, indicating a successful bias reduction. But of course, maybe we just remove bias, but now the predictions is just worse than it was before. You could have like same quality, the same predictions on domain and whole sequence by just predicting everything 1, for example. So we wanted to see if the predictions were still of a similar quality to the ones that you would obtain on ESM, the original checkpoint on the full-length protein. So that's what we did here. So we compared the predictions using the whole context for the same domains using our fine-tuned model and the original ESM. And there are some discrepancies on the predictions, but in overall, qualitatively, the predictions are of the same quality. And to make this more extrapolable, and instead of just training for one epoch, we are using this metric now to decide when to stop training. So essentially, we do the same approach, and when the model starts to perform differently on full-length proteins than the original ESM checkpoint, then we stop training. And we tried this for several domains found on these massive macroproteins. And here is the summary of the results. So in blue, you would have this distribution of discrepancies in log2 scale for all of the domains that we checked. And as you can see, for all the cases, there is some skewness. So there is some under-prediction. So the box plots are more towards the positive values. But after the fine-tuning that we do, then the distribution gets much narrower, and the average also gets close to 0, showing that both the predictions having full length of only domain are now similar, showing successful bias reduction. But of course, here I'm showing that we can reduce the bias, but why is this important? Is this important for any downstream tasks, or is just something cool that we can show on a box plot? So for the downstream application that we are looking at with variant effect prediction, we're using a very simple approach, zero-shot inference, in which we have the wild-type sequence, the mutated sequence, we mask the position, and then we just use as a score the log2 between the ratio of the likelihood of the wild-type and the mutant. And this is a similar approach that has been used before with ESM and alpha missense, so we use this approach on part of their pipeline. And to test these variants, we didn't want to use only clean bar data. So together with experimental collaborators, we curated a more confident data set for which we know and we have a high confidence that these variants are really pathogenic. And for the variants that are not pathogenic, we have also high confidence that they are benign, just to test the model. And here there are the results. And what you can see is that the original ESM was performing quite well. So it was able to separate between damaging and non-damaging. But our fine-tuned model managed to separate a little bit better. So now the boxes get a better separation, and this can also be seen in the performances here in the right. So if you compare the first line, which would be the original checkpoint, to the third line, which is our fine-tuned model, it improved on the different metrics, and it has a similar accuracy to alpha missense, showing that this bias reduction has the potential to also improve other downstream applications, in this case, missense variants. And this brings me to the end of my presentation and conclusions on the future work. So hopefully I'm able to show that for the case of domain sequences, these large language models can have a bias on under prediction for the domains, that you can mitigate this bias with a short fine-tuning using domain sequences, and that this has the potential to improve your downstream tasks. So if you can use the full context, by all means use the full context. But if you are trying to deal with a protein like this, maybe doing an approach as suggested here could improve other downstream tasks. And for the future work, we are trying to make a more general model, and also to have one for each of these common domains on the massive proteins. And also we are generating new variants for validation. This goes back to the point that was also given on the last presentation of having a very diverse data set. So now that we have all these in silico mutations, we designed a data set of variants that is more diverse than what you would find in CleanVar to further validate if the model was right or not. And yeah, thank you all for your attention and to everyone that was involved in this project. Thank you.
Questions? Volker?
I didn't quite understand. So you trained, every refinement is done on all the big proteins, and then you applied it to a data set only of the big proteins or the entire protein genome?
No, so the fine-tuning is done on domain sequences from whole Interpro. So we downloaded all the sequences that had those annotations, chop them up, remove redundancy in a similar way that UniRef did, also clean low-confident proteins, and then train on those. And we were also very careful to avoid data leakage, so anything that was too similar to the massive proteins was removed. And all the results shown are also held out data from some of the clusters that were separated before the fine-tuning.